Hi, everybody, and welcome to Neuroscientists Talk Shop, UTSA's Neuroscience Research Podcast. Today is October 9th, 2025, and we're talking with Maria Geffen. Maria is professor in departments of Otorhinolaryngology, Neuroscience, and Neurology at the University of Pennsylvania. And her lab studies brain circuits of sensory processing and sensory computations in a really general sense, I think including like interactions across sensory modalities that sometimes even appear in the primary sensory pathways and are a little bit of a surprise, and also computational components of behavioral measurements and electrical measurement of neuronal firing and optical measurements of neuronal firing and cell type specific interventions in the circuit to try to sort out the rules governing circuit function. So, hi Maria and welcome.
Hi, thanks for having me.
Also with us is Alicia Bertero.
Hello.
Alicia is an auditory system neurophysiologist in our department and a podcast veteran. You've been on the podcast a couple of times before. So, hi Alicia.
Hello.
Okay, good. And also Alfonso Apicella. Alfonso, if you watch the podcast, you know Alfonso because he's our favorite auditory cortex neurophysiologist and a veteran. How you doing?
Good. Hello.
Okay, good. And also Jon-Paul Muller,
Howdy.
who is a student in our PhD program and an auditory system aficionado himself. Hi.
Hi.
And me, I'm Charlie Wilson. So Maria, I know there are a lot of different things we could talk about. I am open to any topic that any of you want to talk about, but I'd love to start by getting us all sort of focused on the computational function of the various inhibitory interneurons in the auditory cortex as a start. So famously, there are several different inhibitory cell types in the cortex, each one of which is kind of known by the name of some this secondary peptide or protein that it makes that we don't pay very much attention to most of the time. We use it to mark those cell types and we call them by those names. It sounds confusing because we call a VIP neuron a VIP neuron, but we really think of it as a GABA neuron that just has different connections than other VIP neurons. So anyway, we're going to be doing that. We're going to be using that kind of terminology. And what we think is really important about them is not this genetic tag that we use to identify them, but is their connectivity, the particular inputs they get, the way they distribute their synapses on other neurons of the cortex, and the functions that they have in the computational, in the circuit, which we are trying to find out. It's easier to learn about the cells than it is to learn about their computational functions. So let's start with the easy stuff. So let's just spend a minute maybe telling us about the players.
Yeah. So a lot of my research focuses on trying to identify microcircuits in the mammalian sensory cortex that support different types of computations. So typically when we first learn about neurobiology and we study how the brain works, we really think of excitatory neurons. So the neurons kind of connect to each other. There is a cell body, there is the axon, the neuron downstream has a dendrite. This neuron fires, which means it makes an action potential. The signal propagates down the axon, and then through the synapse, it releases a neurotransmitter and activates the downstream neurons. So if this neuron fires and this neuron is connected to it, this neuron eventually will fire as well. That is excitatory neurotransmission. But importantly, if our brain only consisted of excitatory neurons, we would have no way to get rid of extra information or to prioritize important information from non-important information. So, and we would very rapidly become overwhelmed. So and that's very clear in the auditory system in particular. As you're walking down the street and somebody is calling your name, at that moment you want to be able to discard the information from the ongoing traffic, from the natural, from all the natural sounds, from other people talking. You want to filter out that information. You want to concentrate on where the sound of your friend calling your name came from. In order to do that, you have to suppress unnecessary signals. And how do you suppress signals? Well, one way to do that is by using inhibition, by using inhibitory cells. So anywhere within the nervous system, you're going to have excitatory neurons, but you're also going to have inhibitory neurons. And what's interesting about the inhibitory neurons is that they're actually much more diverse than the excitatory neurons. So I actually started studying the inhibitory neurons when I was working in the retina for my PhD. So in the retina, we all know we have photoreceptors and we have retinal ganglion cells that send the signals in the brain. But in between, there are actually inhibitory neurons called amacrine cells that sit in between the excitatory neuronal levels in the retina and control how visual information flows to the brain. And that is a kind of motif that's replicated throughout the nervous system. In the mammalian sensory cortex, if we look at the human cortex, some people have counted upwards of 400 different types of inhibitory neurons. What does it mean that they're different types? They are shaped differently, they connect differently, they have different dendritic branching structure. Their synapses are different. The proteins are obviously different. There's so many differences between them. It's actually hard to categorize them as different subclasses. But it's hard to wire up these and create these different types of neurons. So the question that I've been asking in my lab is, what is their function? Why is it that the brain and evolution has spent all these resources and all of this energy to create these different classes of neurons? What does that give us functionally?
So in the auditory cortex, how many different classes of inhibitory interneurons do you like to think about? Certainly not 400 different classes.
Right. So, it would give me a headache it gives me a headache to think of 400 classes of inhibitory neurons. And so, in fact, we can kind of subdivide them into roughly, I would say, 6 dominant classes. And out of those, so in the auditory cortex, let's say 80 to 70% of the cells are excitatory. So 20 to 30% are inhibitory. Out of those inhibitory neurons, 30 to 35% are the so-called parvalbumin positive basket cells. So those are inhibitory neurons, and they're the most common ones. So we lump them, and there's some subtypes of them, but we lump them all into one class, the parvalbumin inhibitory neurons. Then there is the less common class of somatostatin positive interneurons. Out of those inhibitory neurons, so we have 35% PV neurons, then you have 20% somatostatin neurons. Then we have VIP neurons, which are very aptly named, and maybe 10% of the inhibitory neurons are now VIP
because
neurons.
They're so important.
Because they're so important, there's so few of them, but they can do so much, right? So, and then there are multiple other types that you can kind of put in this like very broad, not overlapping categories of neurons. So we have focused on, in my laboratory, on PV, SST, and VIP neurons.
What are the key connectional differences between these neurons that become the starting point for thinking about their function?
Yeah, so the PV neurons target predominantly the excitatory neuron cell bodies. And so we actually think that functionally that means that they're sending sort of broad dampening signals to the excitatory cell bodies. They also populate the layers. They're kind of project very local. They're very local. So they're dense, they're everywhere, in the cortex, but they don't project too far. They kind of project pretty locally, so they affect excitatory neurons that are nearby.
So they inhibit their neighbors.
They inhibit the excitatory neighbors.
With some exception, correct. We have some of the parabolic neurons in the layer
that can
6.
project
They
somewhere
project across
else.
the layer.
Yeah.
These gain control, correct. It was done first in the visual cortex. I think it was Canciani's lab that showed that. And more recently, I think also Jon-Paul labs did that in the...
These are all still narrow. They don't stretch out and go to far distant places in the cortex, or do they?
That is a very interesting question, because we don't only know that goes somewhere else in the cortex. We know also they go subcortical. But in my lab, we don't call them anymore interneurons. They're called GABAergic neurons.
But we usually don't think of them as having a huge field laterally.
Right.
No.
Maybe they have another field somewhere else.
Yes.
Which also happens in the retina.
Yeah.
Can I ask a question on this? This is a point of not necessarily confusion, but lack of understanding. My understanding was that VIP neurons are a subset of something that falls under the serotonin receptor expressing neurons. So where do those sit? Because I know there's like cholecystokinin and a bunch of other ones, but where, as a subset of those neurons, are you not looking at those as a whole, the serotonin expressing one?
So they're the layer 1 neurons, and I think maybe those are serotonin expressing neurons. The VIP neurons, I think there might be 2 subclasses.
Okay.
And they're distinct from these layer 1 neurons.
Okay.
So the VIP neurons, we think of them as being different from other neurons because they don't so much suppress excitatory neurons, they suppress other inhibitory neurons. So they target somatostatin neurons and paravalvular neurons. The somatostatin neurons target the dendrites, the distal dendrites of excitatory neurons.
So they could, you think of the cell body as the kind of final place. The action potential ends near there. If you inhibit that, you're inhibiting everything the cell does.
That's right.
Whereas out in the dendrites, maybe some input gets inhibited and some other one doesn't.
You could actually, if you're targeting the dendrites, then you can suppress selectively a subset of the inputs so essentially you can think of a cell as an integrator of the inputs across the dendrites. So electrical signals are coming in, they're integrated across the dendrites. Now, if you have a selective synapse from somatostatin neurons onto one of the dendrites but not the other dendrite, then there's going to be inhibition local before the signal is combined across the dendrites. But to be honest, we don't have very fine resolution studies to know exactly, if I take one somatostatin neuron, one excitatory neuron, how much of its dendritic inputs are affected by that one somatostatin neuron.
So it's really just kind of a promising notion, because we know that different inputs can end on different dendrites, and then and so there's an opportunity. The somatostatin neurons have an opportunity to be selective.
Right.
We don't know that they really make great use of that opportunity. It's a cool idea for us, though, because otherwise we don't really have a great way to say what would somatostatin versus parvalbumin neurons do.
Yes.
And I think we try to simplify because we look always at this neuron from a point of view of the pyramidal cells.
That's
it's always what they do on the pyramidal cells.
Yeah.
But the inhibition of inhibition is more probably even a larger mystery of what happened in the cortex. Right? It's this is why I was just, you see, there is always, the recipient of all this inhibition is the excitatory neuron. But I think the GABAergic neurons are also screaming there and saying, but yes, I also get inhibition. But I think, I don't know any study so far that really has looked at the inhibition of inhibition in the auditory cortex. I know in other area, So I think the people are looking, for example, how the PARV and the somatostatin neurons also affect VIP neurons.
Yeah.
So not only the VIP affecting, the PARV and the SOM, is also the other way around. And but I think
so. A lot of the connectivity isn't completely known yet, and it's going to continue to get more interesting and complicated.
Yes, as more we're trying to, depend, correct, where we want to stop, correct? If we keep it at the level of 3 class or subclasses, whatever we identify, I think it's simple. But if we keep doing, the morphotranscriptome or whatever it's called right now, I think, yes, it's going to get more and more probably complex. Because I think, I don't think they reach 400 cell, but, people like Gordon Shepherd at Northwestern, for sure he will disagree with us when we think about that only the GABAergic neurons are diverse. He also believed that the pyramidal neurons,
excited neurons are very...
they form their own, connectivity pattern. I don't know when we're going to stop, Charles.
Yeah.
This is, I don't know.
Yeah, that is a problem because
we search for simplifications because the brain is so complicated, and we need to make some simplifications that make it digestible for us, and then we find out how those go wrong, and we relax our rules and assumptions. But of course, one of the most, since you're here, I'll mention it, one of the most famous assumptions has been all of the output of the cortex has to go through the excitatory neurons. So all of the circuitry is just to control those guys. Yes. But now we know that it's not true, that the excitatory neurons aren't the only, the inhibitory neurons may project out also and carry another signal. And we don't know enough to speculate too much about that right now.
No, I don't know.
But we will. We will. So I think this is kind of background material, and I would really like to get to your studies of in which you have tried to get a handle on the sensory function, not just the circuit and the physiological function, but the sensory function of them. Could you summarize a little bit of that? Get us started talking about that.
Yeah. So in the context of inhibitory neuronal function, we've been trying to understand what fundamental auditory computations the different inhibitory neurons can support. And so first we looked at a very basic problem, which is frequency discrimination. So frequency discrimination is if I play 2 notes on the piano or I pluck 2 strings on the guitar, they're going to sound different. And that's because the frequency of the sound is going to be different. As if I were to, let's say I could tune the guitar a little bit tighter so that 2 strings become almost indistinguishable, or at some point they become indistinguishable from each other. That's going to be your limit of being able to tell apart different frequencies. And that limit is different for different individuals. For example, I personally am pretty tone deaf. However, I know people who have perfect pitch, and they can discern very tiny differences in frequency. So we asked the question, well, does this ability to tell apart different frequencies rely or is it controlled by inhibitory neurons in the cortex? And the reason for that is that fundamentally, the excitatory neurons in the cortex have their own frequency tuning, which means that they can be sharply or less sharply tuned to specific frequencies. And that, if you translate that into population activity, that means that there is some that tuning properties determine the threshold for frequency discrimination. We can up or down regulate the activity of specific inhibitory neurons using very clever tools in mice. And that gives us the ability to test this hypothesis directly in a causal fashion. We can go in and ask a mouse to perform a frequency discrimination task, and we can measure how the discrimination threshold changes in the same mouse when the inhibitory neurons are upregulated, or when they're baseline. Or in a different cohort of mice, we can ask, what if we downregulate inhibitory neurons versus at baseline? How does the frequency threshold change? And we found that by just up or down regulating the activity of these parvalbumin neurons in the auditory cortex, the frequency discrimination threshold could go up or down. And that's within the same session, right? So imagine if you could do this in my brain. So we can't do optogenetics in the human brain yet, although people are trying because it's potentially an extremely powerful technology. But if that were possible, if you could activate the PV neurons in my brain, and I were listening to some piano piece, just during that time when the parvalbumin neurons were activated, I would clear the melody much more clearer, or perhaps it would even hurt my ears because all of a sudden I would be really aware of how mistuned the guitar is or a piano is. But on other times, I wouldn't be able to perceive it. So you can really play with perception in that way.
And Maria, when you tune them down or up, it's constantly change the activity across the layer of the auditory cortex, or are layer specific?
So we haven't done that in a layer specific manner, but that's a really good point. So in general, the mammalian cortex is comprised of what we call 6 layers of neurons, and that actually refers to the different excitatory neurons and their shape and their density across these different layers and their connectivity. And so inhibitory neurons are distributed differentially across those layers. And so it's not something that we have focused on, although we have measured it enough to not see very strong effects, but they might still be there. Yeah.
Of course, when we turn on the light to alter these options, it goes ways. We don't know exactly what distance away we are affecting neurons, right? I mean,
so we make an estimate, right? So we put electrodes, we do pilot experiments. So when we do these experiments, we do a number of pilot experiments. So we put electrodes, we put an optical fiber to activate the neurons, we put an electrode, and we keep moving the electrode further and further away,
to the side,
or deeper and deeper, to just measure, to characterize how far does this optical, while pulsing the light, to characterize how far it goes.
Does it stay in one layer? My experience with these light is that it would spill
goes
out.
across. Yeah. So we have an interesting, currently, this is going to be solved soon, but we currently have a limitation. We can either activate a whole brain region, we can be cell type specific, and we can be sub-millisecond temporally specific, which is amazing. But we either activate a whole region, or we can activate an ensemble of, say, 10 to 100 neurons, if we really try. Probably, 1 to 5 would be more realistic. That's not great. That's not a great scale. Either one of them is not great in our cortex. We have hundreds of millions of neurons, right? But the inhibitory neurons function locally, right? So we're definitely, if we're activating the whole population of neurons, that's just not what happens physiologically. While we listen, we're really activating them in some sort of selective fashion. But at the same time, one neuron probably does not matter, or 5 neurons probably do not matter that much in real-time neuronal processing. We rely, because we have 100 million neurons just for the auditory cortex, we probably rely on more than 5 neurons at any one time to represent information.
Okay, so that's what parvalbumin neurons do.
That's parvalbumin neurons.
I think that's what most of our listeners would kind of expect to pop up in neurons, because it's like
Right,
famously what we imagine inhibitory
Sure,
neurons do.
sure.
So how about the somatostatin neurons?
Yeah, well, all right. So then, okay, so somatostatin neurons. So we started studying somatostatin neurons in the context of temporal adaptation. Now, temporal adaptation is very important. As you're listening to me talk, you're actually only processing, a small fraction, like a very small fraction of your cognitive capacity. When you're listening to me talk relies on actually interpreting, like listening in real time to what I'm saying. Most of your brain is either inferring predictable structure in the sound of my voice or making predictions for what you're about to hear next. And really what your sensory system is doing, it's just confirming those predictions. So it becomes very complicated and cumbersome to listen when those predictions keep getting broken. And so that's, for example, why it's so hard for us sometimes, to talk to somebody who has an accent we're not used to, is because it's hard to make predictions about what they're going to say next, because we're not used to the structure of what they're saying. And so that's just part of how our perception works. If you listen to music, with music, the composers are playing with the predictability of the structure all the time. And, there are mathematicians that have dissected, let's say, like box pieces for predictability and complexity, their structural complexity. Of course, rock music takes that to the next level because it makes very predictable structures and then breaks them, but then it breaks them in a predictable fashion. So what we find is that somatostatin neurons are just crucially important for building up these statistical predictions about sounds. And we first showed that with a very simple sound, which is a two-tone stimulus that goes sort of just like toon, 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 toon. It's called an oddball, and the two tones I basically choose the rate at which I present the two tones, and I have one tone that I present 90% of the time and one tone that you almost never hear. And if you listen to me long enough, I'm not going to bore you, but I promise you will get bored. So if you were just to listen to just one tone over and over again, like toon, 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 you would get very bored.
I was already starting.
More sounds. I'm done. And so, but if I then do toon, 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 beautiful, exciting. And that is literally what is happening in your auditory cortex. In your auditory cortex, let's say I start making those noises, and the neurons perk up and they're like, oh, new stimulus, how cool. They respond, and then as I repeat, the response shrinks. And then when I switch to a different sound, that response gets amplified again, but then it shrinks again. So this process we call stimulus-specific adaptation. 95% of the neurons in the cortex do that. And who is responsible for that? Well, it turns out it's somatostatin neurons. The somatostatin neurons are able to provide specific inhibition selective to the overrepresented tone.
How does the somatostatin neuron know this stimulus should be suppressed because it's...
That's a really good question. So we haven't quite solved it, but we have built a computational model of the cortical network that does the same thing. And so in that model, we can show that there is a specific synapse, a specific property of excitatory to inhibitory synapse that allows the somatostatin neuron to basically kind of accumulate its its activity with repeated stimuli, and then the inhibition that it provides becomes stronger or weaker the more the stimulus is repeated.
So is that just synaptic facilitation that we're used to, or is that some other phenomenon?
Yeah, it's a synaptic facilitation, but at a specific synapse.
Uh-huh.
Yeah.
And how this, I'm imagining this in an autological scenario where the wind is blowing and the animal doesn't need to pay attention to the wind every single second.
That's right.
Or when my son repeat "mom, mom, mom" 1,000 times. But there is a moment when I break because I'm overwhelmed. So I'm thinking could be the role of somatostatin neuron in processing disorders or auditory processing disorder, where the sounds are coming. I think autism and attention disorder, where all the sounds are coming and there's no filter for that and there's no adaptation, I imagine. What would be your...
Yeah, so I actually wrote a grant on that because I think that's a key function. So my prediction is that in the auditory processing disorder, the somatostatin neurons are actually dysregulated and you don't have this adaptation. And so the sounds, you're not able to pick up the predictable and reduce the activity to predictable sounds. That's what I hypothesized, but that has not been shown. Yeah.
I hope that I'm not going to repeat what you already said. You say correct that 95% of the neurons in the cortex, they have this wow moment. But that means that also the parvalvum and the VIP are part of this wow moment?
That's right. Okay. So, and that's what puzzled us, because we wanted to, when we were recording, and so now those results are biased towards excitatory neurons, because when we do electrophysiology, we end up biasing the recordings towards excitatory neurons. It's, as you know, very hard to pick up the inhibitor. They're smaller, their action potentials, are a little bit less strong, not as, so, but we wanted to record directly from somatostatin neurons because our prediction was that they should do the opposite. They should not be adapting, but we found that they actually adapt. So there is a subpopulation of somatostatin that does not adapt, that facilitates, but a subpopulation that adapts. And we unfortunately did not have a large enough population of neurons to really put the ones that facilitate into their own cluster. We thought the effect would be much more obvious. But then we realized when we put that into our model, we realized that because these neurons are coupled in this network, they are going to be overall, as the excitatory neurons are adapting, all of the other neurons are going to be decreasing their activity as well. It's just that they don't need to decrease it by as much as the excitatory neurons to be able to provide this differential inhibitory effect.
So I have a thought on this. If we're talking about, in this particular context, we're talking about somatostatin and it's in this unique role that it can play in the computation of these signals, and you are playing, let's say in the oddball paradigm, you have your tone, 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 and we're looking at predictability. It's starting to build this idea of what predictability is, and you get your oddball, and it's like, okay, hey, whoa, what's going on here? But if we step a little bit further back, we were talking about when we are listening to someone speak, we are building this idea of structures, and we are making predictions based on things we've learned forever ago. These aren't things that we are being shown acutely now. Yes. So if you're hearing someone talk and purple rog fribbit, that comes out. That's entirely unexpected, that bit. But we don't have anything, we haven't built up this acute idea of prediction right now in the moment, but there was a prediction that I shouldn't have made those syllables that I just made, right? Mm-hmm. So would that oddball moment be susceptible to the same mechanics, or is that handled by a different set of connections?
So I actually think that's a really important question, because the reason why I think this research is important is because this idea of prediction and detection of the violation and making predictions and error signals happens at all different levels of cognition. So what you mentioned now is, making predictions about what syllables you're going to produce. That probably doesn't happen in the primary auditory cortex. It's probably stored in higher cognitive regions, or maybe, in humans more in like language and speech specific areas. But it's going to rely on similar principles. So similar microcircuits, just for higher level concepts.
Okay, so would the idea then be that if they're working or they can work through similar neuronal pathways, then you could have, say, through either excitation or inhibition of these somatostatin neurons, you could adjust the ability for someone to recognize audible concepts that are coming through in, say, speech.
Absolutely.
Yeah.
Yeah,
Okay,
that would
that's cool.
be the prediction. Yeah.
Before we have to quit, I still want to hear about the VIP neurons. The most important.
The VIP neurons. And there are many people who have been studying VIP neurons more than I have. We've really just beginning to study them. But we studied them in two different contexts. In one context, we asked a purely theoretical computational question. We asked, well, these VIP neurons, because they have this disinhibitory function, what are they doing to the network structure of activity in the cortex? And so if we look at the ensemble of neurons in the cortex, there is this concept that we have in computational neuroscience called population codes. And the auditory cortex is thought to be special because there the population codes are what's so-called sparse. It's different from the visual cortex. In the visual cortex, if I stick the electrode in, I'll hear many neurons fire. I flash, a moving bar, lots of neurons fire. In the auditory cortex, there's just much less responses. The responses of neurons are much more selective, and we call that sparse coding. And the idea with sparse coding, there's advantages to distributed, so in one example, one extreme you have distributed coding, you have sparse coding, and there is advantages and disadvantages to these different types of codes. But what we hypothesized is that the inhibitory neurons are actually taking this population activity from between sparse coding states and distributed coding states, and that's one of their functions. And we were able to show that the VIP neurons can actually take the network, and bring it to a more distributed representational state, whereas somatostatin neurons bring it to a more sparser representational state. And because, as Alfonso says, these neurons have different projection patterns, they're affected by different cortical regions, that actually gives the brain as a whole a lot of control over representation of sounds, or even representation of contexts, because for different types of tasks, different behaviors, you may want a generalized type of network, or you may want a sparse coding network. And so that can be one of the functions of this neurons.
So when would I like my auditory cortex to be sparse coding, and when might I like it to be?
Sure. We can start with generalization. Why would you want to generalize? You want to generalize is, like I say the word neuron, Alfonso says the word neuron, you say the word
neuron.
We sound different, but they all mean the same thing, right? So we kind of want the representation of these very different sounds to kind of converge on the same thing, right? And that can be achieved with distributed representation. But if, say, I want to very specifically discriminate between two different birds, for example, then I want a sparse representation. And in fact, the birds is where the birdsong system is where this idea of spark coding originated for the auditory system. There, the representations are really sparse and very specific to these different songs. So
great. Well, thank you, Maria. That was great. It was really fun.
Good. It was fun talking to you. Thanks for having me. Yeah.
Thanks.
And it was a lovely visit to UTSA today.
Thank you for being here.
Yeah.
Alfonso, thanks.
Thank you, Charlie.
John?
Thank you. I'm glad you said yes to coming over. So
Great. Yeah.
This has been Neuroscientists Talk Shop.